And so you are the founder, CEO of Celiad, so which was previously called Cardius 3 Biosciences. You're based, so you told me exactly 27 kilometers south of Brussels in Mont-Saint-Gilbert. And so you started as a company exploring cell therapy for heart disease. And from my understanding, the data were not as convincing as you expected, maybe, or as at least it's how it seems. And so you pivoted to CAR-T and oncology. You rebranded to Celiad from your logo, to rebranded to Celiad to basically exploit the potential of your cell therapy expertise of running such a large trial, and to transfer that to CAR-T and to make an allogenic CAR-T treatment. And so first question, I think that's one of the articles we wrote. You just announced amazing results actually at AACR. Just back in April, so good phase 1 results. You were able to cure completely a patient, actually full recovery and no need of chemotherapy preconditioning, which is one of the big challenges of CAR-T right now. So can you just expand a bit on that trial just expand a bit on the trial?
Yes, thank you, Philippe. I think it's a bit more complex than that. The CAR-T program was acquired way before we had the results of the cardiology program, and it is all about optionalities and making sure that you have options. And the cardio program was actually not a failure. It was a failure in the sense that phase 3 did not meet its endpoint. Yeah. But it's a failure. Why did it meet its endpoint? It's basically because it was underpowered. We didn't have enough money to run a trial that was big enough to be able to prove that the efficacy data that we have seen was statistically significant. That's the difference. Now, having said that, the CAR-T again is a very different field in the sense that it is the first cell therapy that actually has made it. And we acquired the technology at a time where the science was proven, the CAR-T general field, the science was proven. But then we really foresee that there was going to be a need for a second wave of CAR-T players. And I think this is where Celiad came in. And yes, the first data we have is in AML, but it is very, very premature data. So it is phase 1. It is not a complete phase 1 trial. It is the first signals that we are getting, actually quite earlier in the development path than we ever expected to have. I'll give you a bit of a background. CAR-Ts have never worked, as you were mentioning, without preconditioning. CAR-Ts until 2010 were not working. Exactly the same CAR-T, second generation CAR-T with co-stimulation, they did not work until 2010. In 2010, Rosenberg first and then Carl June afterwards started adding preconditioning. And this is what made CAR-T work. Our preclinical work showed in animals that CAR-T without preconditioning could work, at least in animals. So we started there. And, a bit to our surprise, we saw that in this context, we did get a response in an extremely difficult to treat disease, which is acute myeloid leukemia. Okay. So, but it is the first sign. Yeah. There is still a lot of clinical development, and we need to have the patients to get that development through.
And can you develop a bit more on why it's a new generation of CAR-T you're developing, and how can you reach that non-preconditioning, or what makes it different?
Well, first, I'm not sure today that we will reach non-conditioned or not. What I can see today is that we are seeing. So CAR-T is made of an antibody or a receptor that recognizes the cancer. And there is a signaling domain under that instructs the T cell to go and kill that cancer. And then there's a T cell, which is the killing agent. Now, CAR-Ts until now have only worked in two types, in B-cell malignancies in general. BCMA type CAR-T for multiple myeloma, or CD19 for ALL and DLBCL. Those CAR-Ts are great. They're providing great benefit at a great expense, both financially, as you all know, but also from a safety and from an efficacy perspective. We think we can do better than that. Yeah. We think that so the next challenges are which one? First, can you make it broader than B-cell malignancies? So we're working on that. Second question, can you make it to work in solid cancers? Very complex target as well to do. Can you reduce the toxicity? And then can you make it more feasible, just from a logistical perspective? So those are the challenges that lay ahead of us.
Yeah.
And
How do you?
So actually we're tracking them through not a single way of doing it. So preconditioning is one way.
Mm.
When you develop a program, sometimes you get things that you do not expect. Frequently you get things that you don't expect, sometimes bad, sometimes good. In this instance, we got something that looked as a response, as a complete response in an AML patient without preconditioning. So what do you do with this? Either you say, okay, it's an accident, and I continue my development plan without thinking of it. Or you start thinking, well, maybe it's an accident, but if I can repeat it 2 or 3 or 4 times, then it's not an accident anymore. And if it's not an accident, it opens up a new patient population that cannot have preconditioning. So we are continuing our dose escalation, and we'll see if we get those 2 or 3 or 4 more patients. But the main path is still testing preconditioning. How to reduce toxicity in the preconditioning approach are other topics that would be too complex to discuss here. How do you address solid cancers? It's, again, people think you have a good target, you will hit solid cancer. No, solid cancer requires a good target. It also requires a good T cell. So that T cell can go and infiltrate the tumor, can deal with the checkpoint inhibitors, which all CAR T today do not do. So you have to think about engineering the T cell to do that.
Yeah.
So it's a early and broad effort that we're undertaking.
Okay. And so on the technology yourself, I mean, you licensed, I think, from Dartmouth in the US, and you licensed out licensed to Novartis. You did a license agreement with Novartis. What attracted them so much in your technology when they already have like a CAR T program on the market?
Well, I need to explain 2 things. What we gave to Novartis is a freedom to operate. We did not give them technology.
Okay.
What we gave to Ono Pharmaceutical is a technology. So it's 2 different type of deals. With Novartis, the freedom to operate is because they wanted to develop allogeneic CAR Ts, and they knew that they need to obtain a freedom to operate, a license from us, to be able to use their technology in this field. So they signed a deal with us, non-exclusive deal with us. Ono Pharmaceutical was interested in the NKG2D based platform in an allogeneic way. So we signed a deal with them to develop the program in Japan, Korea, and Taiwan. So it's really 2 different type of deals.
Okay, makes sense. And on still, how much can you talk really about the gene editing, or how the tech behind that makes your approach allogeneic versus the autologous approach?
So we just presented it 2 days ago, 3 days ago, at the Congress of the American Society of Gene and Cell Therapy, and it attracted a lot of attention because gene editing is always a bit messy. You have to deal with modifying a genome, which is a challenge in itself.
Using CRISPR or TALEN.
CRISPR, TALEN, Megatal, zinc finger. I mean, those are basically 4 technologies that exist today, and all of them have their strengths and weaknesses. And we felt that, and then once you have done that, you need to multiply the cells to a great extent, so you can actually have an allogeneic product that can use the efficiencies of an allogeneic product. Problem is that T cell gets exhausted. You multiply them a lot, they just get exhausted, so they're not effective anymore. So we say, okay, how can we combine this? What they do is cut the genome to take out something called the TCR, the T cell receptor, that is responsible for what is called the graft versus host disease. We said, well, instead of taking the TCR out, we're going to interfere with the signal of the TCR inside the cell, and that's easier to do.
Theoretically,
it's easier to do, but it's actually easier to do. At least manufacturing-wise, it's easier to do. And then when you do it, you find out that you actually, in animal models, that's where you are for the moment, you completely eradicate graft-versus-host disease for a duration of about 60 days, which is what is needed. And you do it during the same transduction step that you transduce your CAR with. So it's not an additional step. The second benefit is because your CAR is a marker, you can positively select your cells. While in a gene editing method, you can only negatively select your cells. You just can basically select cells that do still express the TCR. And the two methods yield very different results.
Okay, makes sense. Well, I guess you could go way more specific, but yeah, we'll keep it here.
Do you have a couple of days? We can.
And so, I guess looking at the CAR-T field, I mean, I guess there's really a lot of hype right now in the field, and not in a bad sense, in a good sense, both in terms of clinical results, which are amazing and has never been seen before in oncology, both in the financial deals, which are just, I mean, talking about Gilead and Kite or Juno and Celgene before. So how are you profiting from all that?
Well, I think we need to look, the way we look at the CAR-T space, there's the generation 1 of CAR-T players, and there are 4 of them. Juno, Kite, Novartis, and Bluebird. That's generation 1. They have data that are getting them to be close to market, or are on the market or close to market. And there's a second generation of CAR-T players, of which we are one, and there are probably 30, 35 companies that are active in that second generation CAR-T player. So you need to differentiate yourself from those 4 and do better than those guys. Otherwise, you just don't have a reason to exist. So that's how we look at it. And we think we're going to be one of those 3, 4, 5 future leaders in the CAR-T space because we have an incredible R&D team, an incredible capacity to invent, and what we are doing, and it will be seen in the next few months. What we're doing, I think, is going to be a platform for all CAR-T players beyond us. So we think, and that's what you need to do when you're second generation companies. You have to really bring something that is of very big incremental value. Otherwise, you're just a me too, and that's not what we want to do.
Okay, makes sense. And I guess a follow-on question still on how you profit from it, but I guess it impacts also the people you can hire, impacts your financial stock, impacts the partners you can attract. Can you expand a bit more on that?
Well, generation 1 validates the concept. It validates that a T cell equipped with a CAR can go and kill cancer. So you start with a level of proof that is already there, and then you need to bring the increment. And so that helps you attract investors that have proven that it works. It helps you attract talents that are excited to be part of this. And it's relatively easy for us. It has been relatively easy for us to raise the funds that we need and to attract the talents that we need.
That's great.
I'm not sure that this is warranted. I think the European Medicine Agency is actually quite helpful and is trying to help companies such as us and technologies such as us reach patients. Country regulations are sometimes a bit more challenging and need to be dealt with. But in general, most of the people we deal with are reasonable. I mean, they want the proof. They want the proof that it works. They want the proof that it's not harmful. And I think it's a fair request to have, to be frank. And the FDA asks exactly the same thing. If by regulatory you mean regulatory agencies, I don't think it's really warranted. Now, if you're thinking about the investment community, if you think about those type of things, that's a different question, of course. First asset that we have is a very broad asset. It's an NKG2D based CAR, which combines innate immunity and acquired immunity, which is already in itself quite unique. NKG2D targets both solid and liquid tumors. So you start with already a target that in itself is very exciting. The challenge with that target is that as it targets stress versus tumor antigen, you could potentially have what is called on-target off-tumor toxicity. So you could have a stress cell somewhere targeting the T cell. So that's why we're looking at it from a very cautious way, and that also brings a lot of challenges from an R&D perspective, to your point, because you're discovering a completely new field. It's totally new. No one has ever targeted stress ligands. So why is R&D exceptional? It's because once you have the target, which is something we acquired, you have to deal with all the problems that that target brings. And you have to deal with, okay, if you want to go after solid tumor, is the target enough? And we don't think it is enough. We think we are seeing some signs today that the target works. But in order really to have a tumor shrink and go away, you need to be much more aggressive against the tumor. Because the tumor has great ways of defending itself, from tumor microenvironment perspective or tumor infiltration perspective. So you have to find those solutions. And this is biotech, to Edwin's point. Biotech is a great place to invent because this is exactly, you get the resources. David Gillum, who's the head of our R&D, has been in academia for 25 years. He said, I've never been able to do as much as I can do here. Because we give the means, we give a purpose, we give a focus. We say, this is what you're doing, this is what you're not doing. And we have a great team of people. Yes, on most of what we're doing, so NKG2D, etc., all of that has complete freedom to operate. But we also have, for example, a CD19 CAR that we are developing. Why are we developing it? Because we think Kite, to give you an order of magnitude, Kite is working in non-Hodgkin lymphoma, DLBCL, to be honest, to be simple. DLBCL, they have a response rate of 35% at 6 months, more or less. 35% response rate at 6 months. So that's great for this disease where the response rate was really, really dismal before that. The question is, we think we can do better than that. If you think you can do better than that, you're going to depend to some extent on the technologies or on the IP of the others. But if you bring a value added that takes that 35% to 50%, then IP is not an issue anymore. Because usually what would happen is that the person that does hold that IP for the 35% response will want to acquire that technology that will make that product better. So at the stage of development we are in, we're extremely cautious about our IP, but that's not the main concern that we have for the moment. First, I think it's very difficult to judge on the first quarter sales or the first half year sales.
It was quite bad. It was like 20 million.
Yeah, for a first product that gets into a relatively small market, it's not that bad. It takes time for physicians to be invested, especially in the US. It takes time for reimbursement systems to kick in. It takes time to see, initially the hospitals didn't know where they're going to pay for it and then wait to get the reimbursement, as it was the case for Dendreon, if you remember. They never got the reimbursement. So all those things need to be ironed out. I think it's not a pill. The cell therapy is something that will take time to establish itself. So I'm not too concerned about that. But having said that, there are many challenges in cell therapy, in particular the costs. And if you go after treatments such as ALL, in which you need to treat 2,000, 3,000 patients a year, a cost of $475,000 per patient is probably manageable by the healthcare system. If you go after colorectal cancer, where you're going to treat 100,000 patients a year, you're going to have to find solutions that are better than that, more efficient than that. Not better, more efficient than that. And we need to do that by maintaining our margins, because that's what's funding our research. That's what's helping us help the patients. And we think we can do that. We think we have a role as industry to actually maintain our margin, make the product more accessible to more patients, and being able to continue to invent. And I think the sales they will need a little bit of time for people to get convinced and progressively to get accepted. If I have a kid today, unfortunately, that will have an ALL, now I wouldn't think twice. I mean, that is a product that I would want to give to my child. So I don't think it's a reflection of anything but slow adoption.
But do you have time as a company? Time to face that slow adoption,
Well, I think at the point where we will be commercial, that probably is not going to be a question you're going to ask me. I think it's actually an excellent question. I think, yes, the answer will come from outside of our companies as well, but we have to do our part of the journey. And for example, I can say now that our cost of goods today is probably a third of what the cost of goods of other companies are, because we have focused from the beginning on the process to make sure that the product is going to be cost efficient, more cost efficient. But there's still a long way to go.
And allogenic by nature is more cost efficient than autologous, or in theory at least.
In theory, and I think it will. But we also have to learn a lot there because I think there's still a way to go on allogenic. My short answer is no, and it's not pessimistic. It's just the nature of the insurances, of life insurances, of funds that are so different that they have so much big amounts of money that they can hire experts to do this, to do the work. So I don't think it will be the case. However, what may change is that we have more US investors coming to Europe and putting their money at work in Europe. And that will make a difference. That's my view, at least. Edwin, you have?
No, I think that's a very good observation. I think the success, if you look in Belgium particularly, of biotech, means that more and more US investors are coming in saying, what's going on in Belgium? Why is it going so well? What's special? And they're coming in, investing and making lots of money. And I think that will then generate further success going forward. But I think you're right, the immense money you have in the US, and it's still a cultural, it's an attitude problem. The feeling that everybody can become president, everybody can win if one of them has been, but everybody can really do it. And I think that's something you can't get away from there, which is a great answer. The question, I think, is whether companies like ours in the future will go public here in Europe first or go straight to NASDAQ.
Or in Hong Kong.
That will make more difficult raising money if you go to Hong Kong afterwards.
No, but they just opened, or Hong Kong just allowed IPOs from biotech companies, and then previously, so now you have some US companies raising on Hong Kong.
But when you're worth in the US, let's say investors will put a price tag on you at say 500, and Hong Kong will put a price tag on you on 2 billion. And then when you're going to go back to US investors and raise money, they won't give you money at that level. So I think that's a challenge as well.